Thank you for tuning in to learn more about the science driving the Atlas PST from Stasys Medical. I am Matt Sobo of Diapharma. Please visit our website at www.diapharma.com for more information. To get in touch with us, please send an email at info@diapharma.com and we'll answer any questions that you may have. So what is hemostasis? Well, basically it's the balanced interplay of forces that act to keep blood within a damaged blood vessel. It's comprised of three non-sequential phases: the vascular spasm, which serves to slow the flow of blood past the wound to increase the odds of platelets and coagulation factors binding to their receptors in the area of the wound. Primary hemostasis is the formation of temporary platelet plug that reduces or eliminates blood loss. It also acts as a scaffold for secondary hemostasis. Normal wear and tear of the blood vessels are repaired exclusively with this mechanism. Secondary hemostasis, on the other hand, is the formation of a stable fibrin polymer mesh. This will eliminate blood loss and entirely seal the damaged vessel walls. When I say non-sequential, it means these all occur at the same time. Coagulation doesn't wait for a platelet plug to be entirely formed before it begins, while vascular constriction occurs throughout the entire process. Here, injury occurs, disrupting the blood vessels and exposing extracellular collagen to the vasculature. Von Willebrand factor, which is constitutively expressed by the endothelial cells and released by the white Pallidi bodies, bind to the exposed collagen. Von Willebrand factor can form very large multimers, which help catch passing platelets at the GP1B receptor and bring them to the basal membrane, where they adhere to the exposed collagen at the collagen receptors. Once bound to collagen, the platelets mobilize calcium ions, activates GP2B3A receptors, emergent receptors, change shape to include lots of pseudopodia to maximize its surface area and secrete its granules. Among the molecules secreted with the various granules are ADP and thromboxane A2. ADP will attract and bind to the P2Y1 and P2Y12 receptors on additional platelets, while thromboxane A2 will bind to the TP receptors on these additional platelets and assist with aggregation. Either of these will lead to activation of additional platelets, mobilizing calcium, degranulating the platelets, and activating GP2B3A receptors. Fibrinogen will bind to these receptors at multiple platelets, anchoring them together to form a platelet plug. Next, actinomyosin and the filopodia contract on the fibrin fibers, shortening the gap between platelets in a hand-over-hand mechanism. This compresses the clot against the tear, allowing blood to flow past the injury. Now, there are many different disorders of the primary hemostasis system that can occur that will lead to excessive blood loss, especially during trauma, surgery, or in some cases of menorrhagia. And also on the other end of the spectrum, improper clot formation, which is no loss of blood, especially those that form in the coronary and cerebral arteries, leading to myocardial infarction or ischemic stroke. The Atlas AST is a direct platelet function analyzer, which measures platelet function without the use of exogenous agonists such as ADP or arachidonic acid. This assay takes 5 minutes from start test to the end of the test. They are accurate in that the contractile forces are quantitative and are measured in the SI units of newtons. The data on the output is relevant. The hemorrhage and clotting risk assessment are both based on physiologic activation of platelets by shear forces. The array of staggered sets of blocks and posts are used to measure these platelet functions. So, how does it work? A small volume of native or anticoagulated whole blood is pumped through the system. Shear forces are generated at the blocks and posts, mimicking what is seen in the damaged vasculature. These shear forces stimulate platelet adhesion to the blocks and posts. As time progresses, these adhered platelets activate and attract additional platelets to aggregate, both to the block and post or to the established platelets. As the platelets aggregate, they begin to exert contractile forces on the fibrin fibers. This leads to the bending of the post, seen here as a shift in the fluorescence signal. This is an image of the block and post pre- and post-platelet. The primary data output in this system are forces measured in newtons and the clot size. This can be seen using the platelet inhibitor. MHP is a metabolic byproduct of clopidogrel, which interferes with ADP signaling. A loss of both clot size and force is generated with increasing concentrations. From these data, additional parameters can be derived, with maximal forces generated, time to activation, how quickly the forces are generated. The dynamics of clock growth, including its stability, durability, and rate of breakdown, can also be derived. Patterns can even be seen when merging these two data sets onto a single plot of forces versus clot size. In this example, you can quickly see that an injured subject suffering significant blood loss shows smaller clots than healthy that are generating similar forces. A severely injured subject, however, both the clot size and the forces generated are diminished significantly, speaking to the loss of hemostatic function seen during severe trauma. A track such as this could indicate the need for a platelet transfusion to prevent exsanguination. Utilizing machine learning, the system analyzes the changes in the output over time, in this case intensity and location of fluorescent pixels corresponding to the block and post, to determine platelet functionality. The example here, high platelet functionality indicates that the subject will likely not need a blood transfusion. While there are undoubtedly many potential uses for this technology, some more obvious uses are in transfusion studies, the testing of platelet potency, both for donated blood as well as manufactured platelets, studying the kinetics of various anti-platelet therapeutics, and also for primary hemostatic disorders. Another interesting use would be in studying complications that arise due to various infections and sepsis. As we are all well aware, many bacterial or viral infections seem to lead to hemostatic defects, even if the agent itself does not appear to interface with that system. I want to thank you for tuning into this presentation on Statlas Medical's Atlas PST and the science behind their technology. If you have any questions, please do not hesitate to reach out to any of us here at DiaPharma by emailing us at info@diapharma.com. Please visit our website, www.diapharma.com. Thank you.